Alrighty, we're going to be going into the last part of chapter 12 today. So let's start off with 12.7. So we're looking at speciation. Um, Currently, estimates are that there's anywhere between 5 million and 100 million species on Earth. Anytime we have a speciation event, anytime speciation occurs, we see one species splitting into two new distinct species. So anytime we're referring to speciation or a speciation event, we're seeing one species split up into two. So one group of organisms splits up into two groups of organisms. And we see two different phases of speciation anytime these events occur. First up, we see reproductive isolation, so they can no longer have offspring, and then afterwards we have genetic divergence. That's just where changes accumulate, and eventually they become genetically quite different. So some organisms have come further along with genetic divergence. That's generally a time factor thing. So as we talked about, the non-reproductively capable hybrids like ligers, like zebra horses, zorses, where they can have offspring but they're genetically infertile. So those organisms have gone through reproductive isolation where they are new species. They are distinct species. Zebras and horses are distinct species. Tigers and lions are distinct species, but they haven't gone through enough genetic divergence to completely make them incapable of having offspring with one another. Just enough to prevent them from merging the species again. We can't have a hybrid of horses and zebras just because enough differences have accumulated to where their children are infertile. All right. Let's look at. We're gonna be looking at phylogenetics coming up. So phylogeny or phylogenetics is the study of the evolutionary history of organisms. Systematics is where we are classifying them out based on common ancestry. So these two things together are how we are assembling the tree of life as biologists, how we are actively trying to sort all the organisms that exist today into categories. And this is something that is very much still underway because tools with genetics are a more recent development. We're getting a lot more accuracy with these trees, with these phylogenetic trees. But it's still something that requires a lot of work, and just because of how many different species, as we said before, anywhere from 5 million to 100 million species, and we don't have the resources set aside to just take DNA samples from every living thing and sort them all onto a tree. So this is, and even if we did have genetic samples of everything, it would still take a lot of work, a lot of math, a lot of effort to sort all those species into a tree. So this is something that is getting better and better and better the longer we work at it. But species continue to change right in front of us, and we still have a long way to go to having a complete tree of life. All right. So phylogenetics. The phylogenetic trees look something like these. So these are some examples of phylogenetic trees. And keep in mind that these are not permanent. These are subject to revision. Very much these things are certain breakpoints we're pretty confident of, but especially when we get closer to species levels, so earlier or more recent divergences, a lot of times we are altering around more recent divergences. This is another thing where also where asexual reproduction throws sort of a wrench, because defining these speciation points where these traits separate us is something that is more difficult to do when we don't have sexual reproduction, when we don't have traditional reproductive isolation. So these are things that we constantly have to think about for asexual organisms. But yeah, you can essentially think of a tree like this as a hypothesis. This is scientists' best educated guess at how these organisms fit together, and then we can reinforce that with evidence or break it down with evidence, depending on whether or not it was a good hypothesis to begin with. So a lot of these have been hypotheses that have been hypotheses that have been well tested as well. But here is so this example right here is just shows that the sort of the order doesn't matter, like which order they are on the top doesn't matter. What matters is where these breakpoints are. These are the key features. So if we move the humans to this side over here, like they show over here, it's still an accurate, it's still essentially the same tree. It's just the how they decided to display it is differently. So that branch point still separates humans from rats and mice. So yeah, just keep in mind that these tweaks can occur. All right. So these phylogenetic trees, these evolutionary trees, show ancestor-descendant relationships. Each of these branch points is called a node, and keep in mind we can spin nodes. So this archaea could be all the way over here. The reason they have it organized this way is because they're showing stepwise where these branch points occurred. So these branches occur from earliest branch to latest branch. So these breaks occurred later in the evolutionary history of life on Earth. And this tree of life is a hypothesis about the breakpoints of all the major groups of organisms we see today. So bacteria, archaea, and eukarya are the three domains we're going to be talking about today. And we're only going to be talking into that highest level because, at least for a start here, just to give you just to sort of dip your toes into the breadth of diversity we see on Earth today. But yeah, we can have several classes about just the differences between eukaryotes. Obviously, the differences between protists, plants, fungi, and animals are extreme. All right. So, what is a monophyletic group? So I might be using this term later. I want you to be familiar with it in case I do use it. So a monophyletic group is something like birds and crocodiles. So birds and crocodiles over here are a monophyletic group. Everything in this box for monophyletic there is monophyletic. So birds and crocodiles, they have a common ancestor, and that means we can classify them under a monophyletic group. However, if we try to group crocodiles and lizards together, we don't have all of the members of that common ancestor. So if we wanted to have a monophyletic group that had crocodiles and lizards, we'd also have to include birds. So this whole thing here, this would be a monophyletic group. But just crocodiles and lizards is not, because it doesn't have all of the offspring of that common ancestor. So we have a common ancestor back here. It splits off into birds and crocodiles, and then off into lizards, it splits off into the common ancestor of birds and crocodiles here. That later diverges again, but this is the separation. Oops, sorry. This is again the separation into two different groups. Remember, every time we have a speciation, we're separating into two different groups. Sometimes that group changes more, so we see birds and crocodiles changing more, and sometimes they don't change as much. So we didn't have any larger groups coming out of this speciation event. But yeah, just keep that in mind. Monophyletic group is where we're containing all of the organisms from a given common ancestor. So that's going to depend on which common ancestor we're looking at. So a monophyletic group for this common ancestor would be birds and crocodiles. A monophyletic group for this common ancestor would be birds, crocodiles, and lizards, which is everything. And a monophyletic group for this common ancestor would be this whole darn tree. That would be our monophyletic group. So how useful it is to call a group, like to assign out a monophyletic group, depends a lot on how recent that ancestor was. Remember, as we get closer to these, the boxes at the top, this is getting more recent. So this is going further back in time. The further down we get when we're looking at these trees, and the further up we're getting, the closer we're getting to these boxes, the sooner in the evolutionary timeline. The same goes for when we go back to this group. These divergences happened very early in the history of life. These divergences, as we get closer to the top, are the ones that are more recent. All right. So one way that we can construct these trees, so constructing these trees is kind of a challenge. It's sort of like a puzzle to think about. One way we can do it is looking at features. So we can do comparative anatomy. Comparative, we're comparing anatomy. We're looking at their anatomy, their physical structures, their bone structures, their traits. We're looking at their phenotypes, their outward appearances. So does the group have retractable claws? That can be our first thing that separates them. Yes, no. So African lions and spotted hyenas have retractable claws. Gray wolves and black bears don't. So we can separate them out that way. Number of chambers in the ear bone. We can separate them out that way. And do they have a penis bone? We can separate them out that way. And those are all three of those structures are structures that we could see with skeletons. And that is how most of our evolutionary trees, our phylogenetic trees, have been determined for extinct animals. So things like the dinosaurs. The only reason why we have trees, evolutionary trees constructed for dinosaurs is using comparative anatomy, using fossil evidence. We don't have any other evidence to use. All we have left is those skeletal remnants, those fossils. So comparative anatomy is something that is very helpful especially when we're looking at extinct organisms, but it's also sometimes used for looking at organisms that are alive today. It is a little bit more difficult to do, and it's potentially less accurate because of reasons we'll go into later. So generally today, if we're trying to determine species, rather than using comparative anatomy for living species, we will use DNA evidence. So if we have a DNA sample from two organisms, we can see how genetically different they are. So based on how much DNA, we can look at specific key genes, and depending on how much those genes have changed, we can determine how closely related they are. So based on the A's, T's, G's, and C's, we can determine sort of how different they are, and where they have speciated. And specifically, a cool thing about DNA too is because certain DNA sequences, I don't want to get into the specifics because again we could spend 2 weeks talking about the specifics of this, but essentially what's happening here as well is depending on how many changes have occurred, we can determine how recently those species have diverged. At least we can estimate that. So we can estimate whether or not it's been several generations or whether it's been hundreds and hundreds of generations since they had diverged into different groups, just based on the number of differences there are. So we can determine how far back in the past. So you see this speciation event is further down. That means it happened earlier, so further back in the genetic or in the evolutionary timeline, and this one happened more recently because it's higher up. That's how we're classifying this again. So if you have a genetic sample, genetic sampling is a great way to determine how these organisms fall into a evolutionary tree. And here is part of why it's hard to use comparative anatomy. So going back again, comparative anatomy, we're looking at similar structures, right? But one thing we talked about when we talked about wings between dragonflies and bats earlier, let's bring that example back again. Here we have the evolution of wings. We have wings coming up twice, once in bats when we get all the way over to here to mammals, and then once way the heck back here, and we have insects developing wings. And we even see different events of insect wing evolution occurring, where completely separate groups of insects have developed flight separately. So remember that similar structures don't always reveal common ancestry. So these are what is certainly, if we go back far enough, they do have a common ancestor. This is not where wings developed. Wings developed later on over here, or over here. And same for insects, they developed somewhere off over here. So they developed this trait independently. So we can't use that as a determiner for how closely related they are. And that is called that is an example of convergent evolution. So analogous traits, again going back to those keywords, analogous traits are ones that have similar function but don't have shared background. Homologous features are the ones where they have the same evolutionary background. So the example there being an analogous trait would be a bat wing to a dragonfly wing. They have similar functions, but they have a separate evolutionary background. So this is an analogous trait. Bat wings and insects wings are an analogous trait. But if we look at bat wings compared to our hands, we see a lot of homologous features. The bone structure of a bat's wings are very, very similar. So they have their arms that sort of are the structural support for their wings. All the bones in their arms have homologous bones to our hands and our arms, and that's because we're fairly recently diverged from them. So that is an example of a homologous feature. So when we have the difficulty of determining, well, is this an analogous trait or a homologous feature, one of the easiest ways to determine whether or not it is by using DNA analysis. And another example of where you might get thrown is sometimes they just even look similar, just because the body plan that is successful in one area might be successful in another area, and evolution will just sort of push organisms to look similar. So here we have a burrowing organism, a little mammal, a little rodent that has figured out, or that has been successful digging up ground-dwelling insects and eating them. We have another example of that happening over here, and they have developed very similar features, but they have completely, a lot of those features developed completely independently. And we see, in fact, that these guys are very genetically different by comparison. So African golden moles are more closely related to African elephants than they are to the common shrew. And that's something that is very difficult to do with comparative anatomy. It might be possible with comparative anatomy, but it would be very, very difficult to do. But it is quite easy to do with DNA analysis, with genetic analysis. All right, it's important to think as well about the levels of evolution that are occurring here. So we have different kinds of evolution, different classifications for evolution. So let's look at micro and macro level evolution. So microevolution is where we're looking at just over a few generations, and we can have a lot of differences occurring over a few generations. That's specifically we're looking at like one line. We're looking at the changes in allele frequencies. Ooh, my slides. Sorry, this got cut off here, and that's my bad there. So changes in allele frequencies over one or a few generations. I'll have the slides reformatted and posted later. That's my bad. So again, changes in allele frequencies over one or a few generations. Macroevolution is the accumulated effect of microevolution over time. So when we look at the over a longer period of time, we see a lot more changes, a lot more speciation events. So in this case, we have this one group leading to a speciation event here, and then another speciation event here. When we're just looking at the changes over time that lead to this group separating off into two different groups. That is microevolution. But when we look at multiple speciation events, when we're looking at many generations, that's where we're looking at macro level evolution. We see microevolution happening all the time. This is something that is a good example of, we can use human epidemiology as an example of microevolution. So part of the reason why rhinovirus, the virus that causes the common cold, is so successful in human populations is because it's constantly evolving. It's undergoing microevolutionary changes. It's changing different allele frequencies, and keep in mind those generations are incredibly quick for a rhinovirus. So yeah, they develop new traits. They go through microevolution. Where we're looking at macroevolution, we would say maybe a good example of macroevolution would be the divergence of the dinosaurs. So we can track all the speciation events that led to the major groups of dinosaurs. That would be an example of studying macroevolution. All right. So another thing is we like to think of evolution as a slow process that occurs over millions and millions of years. But the truth is that that is not entirely the case. So it can take place over many, many, many years, and we do see some instances where we have long periods of gradual change, and this definitely is dependent on organism to organism. So this is not like the whole world at once is going through graduated change or punctuated equilibrium. Different species will do different things depending on the conditions they're under. So gradual evolution, again, change, slow change, incremental. Punctuated equilibrium is we say it's evolution by jerks. It's by sudden changes. So we'll have periods of stagnation, then sudden changes, and periods of stagnation, then sudden changes. And a lot of times, this will have a sort of an event that corresponds to this. So something will change in the environment of the Earth that will cause rapid changes in an organism. And this is actually one of the big things that we see happening around our mass extinction events. So every time there is a mass extinction event on Earth, and there have been at least 5 major ones that we know of, there has been massive and very quick changes immediately afterwards. So every time there is a large die-off, there is a lot of open energy available. A lot of things that used to be getting consumed rapidly are no longer being consumed. A lot of the organisms that used to take advantage of that energy are now gone. So that opens it up to new organisms. Those new organisms will rapidly evolve to fill those gaps, those that openly available energy. We call that a niche, by the way, N-I-C-H-E. So a niche is where we have a sort of a gap being filled. So the niche of crows, I'd say, the niche of crows in the urban environment is that they are excellent scavengers. They're very good at picking leftover food from humans. So that is their evolutionary niche. If they were not doing that job, another organism would very soon fill in their gap. So maybe if crows were gone, then seagulls would speciate more to fill in all the niches that were left behind by crows. And we actually do see a lot of those kinds of things happening where we have 2 similar species that share a niche. If one dies out, the other one will sort of fill the gap and then speciate out again. But yeah. Sorry, going back to the big ideas here, though. Gradual change, slow process, punctuated equilibrium, where we have these big events that change the concentration of genes very quickly and can lead to speciation events. So punctuated equilibrium is that term. All right. So Another thing we see during those big die-offs is adaptive radiation. So specifically, if we have an ecological niche, a free energy, like a free source of energy, like waiting to be exploited essentially by another organism. A lot of times, what we can see is a new organism coming along, and it is separate from the larger population, and it very quickly will reproduce and diversify. And that's something we see in the events that happen around adaptive radiation. So adaptive radiation is radiation doesn't mean like radioactive in this sense. It means to like radiate out from, to move away from. So if I were to radiate out from a group, if I was in a group circle, we were having a chat, and I were to walk away from them, I would be radiating out from the group. That's the kind of radiation we're talking about here. So this is when we like one small group will split from a larger group and colonize a new area and rapidly change. And we see during the most recent mass extinction event, we see mammalians. So our early ancestors were the ones that really were successful at radiating out and creating a ton of different species, and that's why we see mammals all over the earth today, is because mammals were one of the most successful groups at radiating out, at spreading out, diversifying, and consuming up all the new free niches that were left behind by the dinosaurs after the dinosaurs went extinct. So we talked about mass extinction events as causing adaptive radiation, but there's other things that will cause adaptive radiation as well. So we have colonization events. So when we talk about Darwin's finches, though that was the result of a colonization event. A small population just moved to an island. On that island, they're able to move to that island maybe once, twice, maybe a few times. But once they're there, it is very energetically expensive for them to fly back and forth. So they generally don't do that unless they're desperate. And what happens there is we'll have a new population become isolated, and that new population, having been geographically isolated, will eventually become reproductively isolated as well. So that is an example of an adaptive radiation leading to a speciation event. So colonization, and sometimes we have a evolutionary innovation. So one trait, perhaps an emergent trait, a new trait, is so changes the fitness of an organism so much, it changes its ability to reproduce successfully so much, that it rapidly produces an upswell in the populations that share that trait, and sometimes they leave behind a section of the population that does not have that trait. So one population will emerge very rapidly that has a very distinct trait. And when we talked about different types of evolution previously. So this is an example where maybe the heterozygotes of this trait were very unsuccessful, but homozygotes one way or homozygotes another way were very successful. That leads them to sort of separate out, and eventually they become separate species. All right. And let's also keep in mind that we have had, again, several mass extinctions on Earth, at least 5 major ones that we've classified out. Obviously, there have been more events that have caused large amounts of groups of animals to go extinct, or large amounts of groups of organisms to go extinct. But the 5 major ones were the most dramatic ones that we have found. So remember, the sort of the definition of extinction here is the complete loss of all individuals in a species population. So if you lose all your organisms in a group, you are extinct. But yeah, let's look at sort of what's, I guess, some of the history of extinction here. So we have our mass extinction events. That is these huge spikes in loss of life. And we also have smaller background extinction events occurring. So sometimes um, there'll be changes in the environment that lead to large amounts occurring, but then we have these larger spikes occurring as well. And just keep in mind that every time a speciation event occurs, we have one species turning into two different species. So it makes sense that extinction is happening at a fairly similar rate. Pretty much, I can't say at the same rate, but at a similarly consistent rate, we have extinctions happening to speciation events. All right. So about 65 million years ago, we had an event that used to be called the KT mass extinction. Now we call it the Cretaceous Paleogene extinction event. But yeah, this was the event that 65 million years ago wiped out the dinosaurs. It also wiped out 3/4 of all species on Earth. Um, it didn't wipe out. It's important to know that it didn't wipe out all all of the ancestors of the dinosaurs. Obviously, we still have birds today. Birds we now recognize as sharing common ancestors with the dinosaurs. Um, but yeah, we still have a large loss of most of the large scale organisms on Earth. A lot of the larger organisms did not have enough available energy to survive this extinction event. So smaller organisms sort of filled in those niches, and that was our ancestors. So our early ancestors were very, very small organisms. They were small therapsids. But anyways, so I gave a little bit of background on the Cretaceous Paleogene already, but I want you guys to choose one of your own. You can choose that one, or you can choose one of these others. I highly encourage you to look into one that you don't know much about, because that's going to be more interesting to learn about, let's be real. But we've had several mass extinction events happen during the history of life on Earth. At least these 5 major events that we know of. I want you to choose one of these 5 events, and then I want you in 1 or 2 paragraphs, so that's 4 to 8 sentences, summarize what occurred and then write it down in your own words. I want you to sort of let me know what major species, which major groups perished during that time, which species developed after the extinctions, and what, if we know what caused it, what caused that mass extinction event. So I want you guys to look into that. And remember, in your own words, I want you guys to interpret what you're reading and retranscribe it using your own language. And also make sure you tell me where you got your information from. Wikipedia is not a source. Where Wikipedia got their information from is a source. So make sure that you are looking for first-party sources for this. One will do if you can get all the information you need from one. Just make sure you tell me where you actually got that information from. Hyperlinks are good as well. All right, moving on. So I'm just going to have a quick look at all the domains of life on Earth. So earlier on, we had Linnaeus, who classified things into plants and animals, and then he had a mineral kingdom for non-living matter. We have since revised this heavily. We know there's things other than plants and animals. This definition was based on he classified organisms that created energy, so plants, and things that consumed energy, which were animals, but that is an inadequate system. certainly useful at the time, but I'm sorry, I'm going to cough here. All right, I had to mute myself. So it was good for the time, but we've since gone much more advanced. Um, some so an example of an organism that doesn't fit into the two categories, we have like microbes that can do things like animals or like plants. So this classification really had some drawbacks. Later on in the 60s, we had a 5-kingdom classification. 1 kingdom of prokaryotes, 4 kingdoms of eukaryotes. Later on, we revised this again. This is closer to where we are today. The biggest problem with this tree of life is that if we look at where protists are here, here, and here. And that means that we don't have protists as a monophyletic group. They have multiple points of origins. So with protists being separated out from 3 different speciation events, that means the protist group is not well addressed by this. And this was based on the study of RNA specifically. So this was one of the first that was put together via genetic evidence, molecular evidence. Another thing is this was the first tree that classified out archaea. So we have archaea being one domain that is separate from protists, but also separate from bacteria. And we'll talk a little bit more about those today. All right. So again, protists, not a monophyletic group. This is a problem. And another problem is bacteria have what we call horizontal gene transfer. That's where genes can pass from individual to individual within the same species or even between species. So some bacteria are capable of sharing genes between species. That's those plasmids, and that's one of the ways that that multi-drug resistance can pass from organism to organism, even if they didn't develop the drug resistance themselves. Is if we have those genes occurring on a bacterial plasmid, a little circular piece of DNA, that little circular piece of DNA can get taken up by a completely different bacteria, and then it can become pathogenic. It can become dangerous. The other problem with this is viruses aren't included. So the supposition of this tree is that viruses are not living, and that is something that is not hard and fast in the study of biology. Viruses do undergo evolution, we know that, but they don't go through their own metabolism. But not all scientists agree that they are not living. So this is something that it's exceptionally hard to classify. It's very hard to classify viruses because they don't have metabolism, because they only work based on exploiting the resources inside of other cells. It is very, 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 very hard to track individual species or strains of viruses and their evolution. They just change so much that it's hard to track them back. So, let's see. Yeah, this is the most commonly accepted theory of the evolution of life on Earth, which is we have some kind of ancestral bacterium. They split off between bacteria and archaea. So yeah, these groups separated, and then later on they had endosymbiosis. Endosymbiosis was where smaller organisms, so smaller bacteria or archaea, associated within larger ones, and we had this the sort of separation of tasks into different membrane-bound organelles. So things like the mitochondria, the Golgi apparatus, all these things that were membrane bound sort of became these specialized organelles. So that was that speciation event. It was the origin of organelles. All right, so let's keep in mind that we have these three major groups, bacteria, archaea, and eukarya, that we're dividing into. So these three groups do sort of stand up to the rigors of time. Bacteria, archaea, and eukarya is still a very good way of classifying organisms. The issues come when we look at protists. Protists within eukarya, it's very still very much a group that we need to improve on separating out. And keep in mind that when the things that we think about as being the most important, plants and animals, are right here, and that's just two groups within eukarya. So that is something that is only a very small fraction of the diversity of even just eukarya, and especially when we look at all of life, it is a very small percentage of the diversity of all life we see on Earth. So much of the diversity of life we see on Earth is between bacteria and archaea as well. We see a lot of different species in bacteria and archaea. We just tend to favor looking at macro scale organisms, large organisms like ourselves, as being more important because we're kind of biased that way. All right, so let's look at bacteria. So bacteria is a monophyletic group, so it does fall out with one common ancestor. We can at least hypothesize well back to a common ancestor. These are all single-celled prokaryotes. They all go through asexual reproduction. They're classified based on physical appearance or genetic sequence. More often than not, we're favoring genetic sequencing nowadays, especially for these guys, because a lot of them have examples of convergent evolution for similar looking features. And some cause disease, but all of them, but they are also essential to our daily lives. So the ones that don't cause disease are just such an important part of our ecosystems and our bodies. So when we look at our bodies, something that we're growing to appreciate is our microbiomes, the colonization of different bacteria on the surface of our skin and our GI tract. All of our breathing pathways are also colonized by bacteria as well. So it's important to remember that bacteria aren't all bad. We also see bacteria playing a critical role in the process of recycling nutrients back into the ecosystem. So they're one of the things that's helping to break down dead organic matter in the soil. They are helping to they are constantly taking in and putting out things in our atmosphere as well. So keep in mind, bacteria play a huge role in our current ecosystems. So one of the only thing I really want to focus on with this slide is looking at the broad diversity. Keep in mind, bacteria speciated a lot, or the common ancestor between us and them happened so far back in our evolutionary history that, and these things reproduce so quickly that keep in mind that these things will be much, or can be much more genetically different from each other than even different plants and animals from one another. These bacteria have had an immense amount of time to grow very, very, very genetically different from one another. So some of these have incredibly diverse genes and incredibly diverse metabolisms and incredibly diverse reproductive capabilities. Some of these will go through that horizontal gene transfer we talked about earlier, where we have a small section of DNA being traded from one organism to another. Some have other methods of exchanging genes as well. All right, so that was bacteria. Let's look at archaea. So Archaea split from Eukarya. And this is a more recent event than the bacterial split. So again, we'll go back to this split. So this is our first split, bacteria from Archaea. Later on, we have Archaea splitting from Eukarya. So we have these two groups separating from this one group. And let's look at that. That is Archaea split from Eukarya more recently. And then these are all single-celled again. These are still prokaryotes. And a lot of times they will have cell walls that contain polysaccharides that are not found in bacteria or eukaryotes. So they will have very distinct cell wall structures. And we see a lot of these Archaea are extremophiles. So extreme just means, very extreme conditions. Extremophiles means they love extreme conditions. Philes is the root word for love. So thermophiles are heat lovers. Halophiles are salt lovers. We have Archaea that are tolerant to low and high pH, some that are resistant to high pressure, and then some that produce methanogens. Things like when you hear about the deep sea hydrothermal vents, a lot of times we're talking about Archaea living down there. So a lot of these, because a lot of these live in extreme conditions, they're not as well studied as some other organisms. So there's always the possibility that we find completely new and novel things as we begin to develop better tools for studying them. I'm going to push right along to Eukarya. So Eukarya is all these organisms that have membrane-bound organelles. So most are visible to the naked eye, not all, but most. Most are multicellular, and in most species that we have like names for, like common names for, like the baboon, the mushroom, are eukaryotes. Because these are the things that were classified when we had the ability to only see at the macro scale. So remember, this is the key thing for eukaryotes, is that we have these organelles happening within our cells. Plants, animals, fungi, and protists all occur in Eukarya. All right, so moving on. So that is all I got for chapter 12. Coming up is chapter 11. So we're going back in the book to chapter 11. Next reading assignment is 11.1 through 11.9. That's pages 344 through 356. Remember to look to Blackboard as well. So with that, I hope you guys all have a great day, and I will see you all next time.